Welcome to The Explainer. Today, we're taking a massive deep dive into what is arguably the most profound scientific revolution of our time. We are unpacking the mechanics, and just as importantly, the heavy ethical implications of genetic engineering. We're going to look at a medical marvel that's literally allowing us to change an organism's fundamental features in record time. We'll explore exactly how it works on a molecular level, what it can do for our world today, and the immense responsibilities it places on our shoulders for tomorrow. It's a fascinating subject, so let's jump right in.
Okay, let's dive into this syllabus. We'll start with 1, rewriting the instruction manual, followed by 2, how molecular scissors work. Then we'll look at 3, the somatic successes, before upgrading to our ethical discussion in 4, the germline editing debate, and 5, therapy versus trait enhancement. Finally, we'll wrap up with 6, the unwritten future.
Section 1. Rewriting the instruction manual, the basics of DNA.
To truly wrap our heads around the magnitude of this tech, we really need to start at the foundation. This quote perfectly grounds our understanding. The DNA contained in our genes acts like an instruction manual for our cells. From the absolute smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. There are 4 building blocks, called bases, and they're strung together in these incredibly precise sequences. These sequences are the instruction manual. They tell the cell exactly how to behave, and they form the basis for our every single trait. Basically, when you change the manual, you change the organism.
And that brings us to the absolute star of the show, CRISPR-Cas9. I know, it's a huge mouthful. It stands for Clustered, Regularly Interspaced, Short Palindromic Repeats, and CRISPR-associated protein 9. But what you really need to know is that it's a fast, cheap, accurate, and incredibly efficient gene editing tool. But honestly, here's the wildest part. Humans didn't invent this from scratch. There's no way. It's actually a natural process. It's long functioned as an immune defense system for single-celled bacteria and archaea. See, when a virus invades a bacterium, the bacterium captures a little chemical snapshot of the infection, just a tiny snippet of viral DNA, and stitches it into its own CRISPR region. If that same virus ever attacks again, the bacterium produces RNA that acts like a scout, recognizes the virus, and uses a protein called Cas9 to chop up the viral DNA. Back in 2012, scientists figured out how to hijack this brilliant bacterial defense mechanism. They realized they could target not just viral DNA, but any DNA in almost any organism, a real game changer.
Section 2. How molecular scissors work, the lab process.
So, looking at the molecular mechanics of that 2012 breakthrough, here is the step-by-step process they used in the lab. First, scientists design a short piece of custom guide RNA to perfectly match the specific gene they want to edit. Second, this guide RNA attaches to the Cas9 enzyme. That guide RNA acts exactly like a GPS, directing Cas9 straight to the intended target sequence in the cell's DNA. Third, Cas9 acts as molecular scissors. It mirrors the exact process it uses in bacteria and just snips the DNA at that precise targeted location. And fourth, once the DNA is cut, the cell's own natural DNA repair machinery kicks in to fix the break. This right here is the key to CRISPR's immense power. By just injecting Cas9 bound to a custom guide RNA, scientists can edit practically any gene in the genome. It is literally almost as easy as fixing a typo in a Word document.
Now, what's really interesting about this is how we actually manage that final step, the cell's repair process. We have 2 distinct pathways here. On one hand, we have non-homologous end joining. Naturally, proteins called nucleases try to trim the broken DNA ends and just mash them back together. But, this is really prone to mistakes. It can lead to extra or missing bases, and the resulting gene is often unusable and effectively just turned off. But then we have homology-directed repair. If scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins use that template as a blueprint. This allows for incredibly precise rebuilding, meaning we can repair a defective gene or even insert a completely new, customized DNA sequence.
Section 3. The somatic successes. Applications in today's world.
Because we have that kind of blueprint precision, the real-world applications are just staggering. But before we get ahead of ourselves and look at the breakthroughs, we must define the boundaries of our current clinical work. Right now, most changes introduced with genome editing are limited strictly to somatic cells. These are cells other than egg and sperm cells. This is absolutely crucial to understand, because genetic changes made to somatic cells are isolated to only certain tissues within that specific patient. They are not passed from one generation to the next. And that creates an essential ethical safety net for researchers exploring these therapies today. And within that safety net, the potential is absolutely boundless. CRISPR is being explored in research and clinical trials right now to cure single-gene disorders like cystic fibrosis, hemophilia, and sickle cell disease. It holds amazing promise for treating complex diseases too, like reprogramming drug-resistant cancer cells or preventing HIV infection and heart disease. And because DNA is the universal instruction manual we talked about, this isn't just limited to humans. Scientists can engineer drought-resistant crops, create apples that don't brown, or yield much larger fruit. They're even looking at creating mosquitoes that literally cannot transmit malaria, potentially preventing the spread of massive infectious outbreaks.
Section 4. The germline editing debate, affecting future generations.
Now, the heavy ethical challenges really kick in the second we step away from those somatic successes and look at germline cells. These are egg or sperm cells, or the genes of an embryo. So the crucial point is this: genetic changes made here are not isolated to one patient. Changes made to germline cells could be passed from one generation to the next. This completely shifts the paradigm of genetic engineering.
It presents us with an incredibly profound question, one I really want you to think about as we move forward. If we have the technological ability to delete a severe genetic disease from an entire family line, from all future generations, should we?
Like any good classroom discussion, we've got to impartially look at both sides of this. On one side, the potential benefits of germline editing are monumental. We're talking about the permanent eradication of horrific inherited diseases like hemophilia and sickle cell disease before a child is even born. But on the other side of the debate are massive safety concerns. The reality is, CRISPR isn't perfect yet. It doesn't always make just the intended changes. There can be off-target edits and mistakes. And because these edits are passed down, it's incredibly difficult to predict the long-term implications of a single CRISPR edit cascading across multiple generations of human life.
And this brilliantly illustrates how the global community is actually handling this immense responsibility right now. As it currently stands, somatic cell research and lab models are active and legal. Scientists are actively using this to understand diseases and test individual therapies. However, germline cell and embryo editing is currently illegal in the United States and many other countries. The primary reason is exactly what we just discussed, those profound, unresolved concerns regarding ethics and multi-generational safety.
Section 5. Therapy versus trait enhancement, drawing the line.
You see, the safety of future generations isn't our only ethical hurdle. We also have to figure out where to draw the line on what exactly we're allowed to edit. Germline and embryo genome editing forces to contrast two very different goals. For starters, we have therapeutic disease treatment. This is the use of the technology to restore normal function, to prevent HIV, or treat heart disease. It's fundamentally about healing. But then we have controversial trait enhancement. This brings up the massive ethical challenge of whether it's permissible to use this technology not to heal, but to alter normal human traits. Should we allow the engineering of embryos to change a person's height or to increase their intelligence? It's the highly debated difference between treating an illness and upgrading a human being.
Let's move to our final topic and see how this all builds. Section 6. The unwritten future, leaving the lab.
The reality we all have to face right now is that CRISPR is no longer just some theoretical concept. It's a powerful tool actively being used to study the genome and watch what happens when genes are turned off or changed within an organism. It has left those single-celled organisms way behind and is aggressively heading into our labs, our farms, our hospitals, and into organisms all around the world. The technology is here for sure, and it works.
Which leaves us with one final unwritten question to wrap up our explainer today. As CRISPR leaves the lab and enters the real world, raising these massive ethical questions about germlines and enhancements, who decides? It's up to us to decide the best course forward, but who holds the ultimate moral authority over our shared genetic future? Think about that as you consider the incredible power of the molecular scissors we explored today. Thank you so much for joining me on this deep dive. Keep questioning, keep learning, and I'll see you next time.